All right, so we are on to chapter 3. And with chapter 3, we are now moving up that level of organization. Remember, chapter 1 was just introducing anatomical terminology, the basis for homeostasis, and why it's important. Chapter 2 was on the chemical level of organization. Now we're on chapter 3, where we're actually talking about cells. All right, so cells are the basic unit of all living things, and there are characteristics specific to cells. They have these characteristic functions, things that make cells cells. The first one is cell metabolism and energy used. All the reactions that occur within a cell gain or release energy that is not consumed, it is transferred. So energy is transferred, not used. The second thing is communication. Communication happens between cells. No, it's not blab, blab, blab with the mouth, but communication is actually done with chemicals released and detected. Also, electrical signals allow for communication. And again, you know how it is. We're going to get more in depth with this as we go on through A&P. The third one's reproduction and passing on DNA. It's the division of cells containing that genetic material, containing that DNA, and passing the DNA. Think about it. When a cell is fixing to divide, it divides equally. It divides equally of all the genetic material. So there's your passing of DNA. And the last one, synthesis of molecules. Cells within the body produce things like proteins, nucleic acids, and lipids. All right, so we talk about this cell theory. This is the idea of life and its basis associated with cells. So all living things are made up of cells. They're found in anything that is alive. Now the numbers vary, but they are required to demonstrate or define things as alive. Cells are the basic unit of structure and function in an organism. Cells dictate that organism. It dictates how the organism survives, how it functions, what it looks like, because it is the basic unit of life. Like I said on that first slide, this is the basis. This is the beginning of life. Think about it. In fertilization of a human being, the sperm meets the egg and then it's fertilized. That is an egg. That egg changes and divides into many, but there's the basis of life. And lastly, cells come from reproduction. That's cell division. We've already talked about passing the DNA. So cells come from cells. All right, so we're going to talk about the organelles next. I just want to put a picture, which I am sure, or at least I hope, you're very familiar with from even high school, or if you've taken basic biology at our school. Okay, so we're going to start with the plasma membrane. It's a boundary. It is a living layer. Remember, we talked about boundaries being a characteristic of life. It separates internal and external environments. It is double layer of phospholipid. Remember that those phospholipids, the heads are hydrophilic, water-loving, so they're pointing out towards the water, and they're made up of glycerol and phosphate, which you can see the image on the right. The tails are fatty acids. They're hydrophobic, so they're face inward. So look at that depiction at the bottom, that image at the bottom. You can see the heads pointing outward. That would be the barrier, if you would. That would be what is in contact with the watery environment. And then you can see the hydrophobic tails on the inside. This plasma membrane laterally move to allow small molecules like oxygen, carbon dioxide, and water to enter. Okay? These are found around all cells. And let's see, what are the components of it? Okay, the components of it are going to be the cell membrane proteins. Membrane proteins help move large molecules or aid in cell recognition. So small things, like I said, can move in and out of the cell. However, there are big things that do need to move, and proteins add a channel, if you will, to move things in and out, which you can see the protein channel. Those allow for bigger things that are needed within the cell or things that are synthesized and need to come out of the cell. Okay, there are two types of proteins. You have peripheral proteins, which are attached to either the inner or the outer surface. You have integral proteins. Think about it. To integrate, integral proteins are embedded through the membrane. And lastly, you have glycoproteins. They have carbohydrate tails to act as markers for cell recognition, which are represented in the blue on that plasma membrane. The idea is cells have to be able to recognize cells, which we'll talk about immunity in the future, but your body, if it doesn't recognize something as self, then it will quote unquote attack it. It will produce antibodies against it. So these glycoproteins with the carbohydrate tails allow your body to recognize that cell as self. That cell membrane is fluid. I told you it moves, and it is constantly in motion, so it has fluidity. Also, it presents this mosaic model. Now, to think about a mosaic model, yeah, you know those Spanish tiles, or if you look at somebody having tiles, whether it's in the bathroom or outside, and they've got a beautiful pattern to it. So if we were to take the plasma membrane, and instead of looking at it horizontally, you look down onto the surface, you would see this pattern to it, hence the name mosaic. Now let's move inward and start talking about the organelles that are present. First is cytoplasm of a cell. It provides a medium for all chemical reactions. Like I've talked about before, you need a fluid, if you will, containing water to allow for chemical reactions to occur. If there's no chemical reactions by that cell will not survive. We also find organelles present in it. They float, if you will, within it. And cytoplasm is found within all cells. Now, the best way I can describe what this cytoplasm is, because you can't touch it, so you want to kind of get this in your mind. So let's say you are on an all Jello diet. Man, Jello takes about half an hour to completely solidify. And this all Jello diet, you are starving from breakfast, you have not had it. So you make Jello, stick it in the refrigerator, and you come back in about 10 minutes. It's not completely liquid yet, but it has some depth to it. It has some density to it. It's not completely solid, it's not completely liquid. Imagine touching that right now. That's what cytoplasm feels like, if you will. So we're going to talk about next the nucleus. This is the control center for all activities. It's kind of like the brain of it, if you would. It contains the genetic material in the form of DNA. It has it condenses in dividing cells and as chromosomes, and then it shows as chromatin in non-dividing cells. Okay. It's bound by a double membrane surrounding the nucleus. Okay, this double membrane is called a nuclear membrane. It contains nuclear pores that allow things to move into and out of the nucleus. And the nuclear membrane connects to rough endoplasmic reticulum. Cytoskeleton, like the name suggests, cyto means cell, skeleton, you know what the skeleton is of the human body, and it's really important. It has some significant functions. Number one, it maintains the shape of the cell. That makes sense. It also moves organelles, kind of like the bones aid in the movement of humans. It's made of protein. And there's two types. You have microfilaments, which are very thin, and they're made of actin, that's the protein. Then you have microtubules, which are thicker. They look like, now they're not as big, but for reference and understanding, they look like a straw. Those are made of tubulin. So microfilaments, actin, microtubules, tubulin. Again, you know it's a protein because it ends in IN. All right, so centrioles, they're only found in animal cells and they're always found in pairs. They have a circular shape, which you can see the image at the bottom of microtubules. So imagine two of those kind of line in different directions. They're involved with pulling chromosomes apart to opposite poles during cell division. And again, if you don't remember cell division, that's going to be the tail end of this chapter where I'll go over it. All right, so the mitochondrion. Flora is mitochondria. Okay, remember that nickname, powerhouse of the cell, plant found in both plants and animal cells. And I know you know that because that's the standard. Hey, powerhouse. Think about it, powerhouse. It provides the energy necessary. So it makes ATP. And this goes without saying. Okay, think about it. The more active the area of the body, the more mitochondria you would find. Think about it. Skeletal muscle, that's what moves us. That's the stuff if I flex, you can see. Skeletal muscles have to or require more energy, so it has more mitochondria present to produce that ATP. Cellular respiration, which we'll talk about shortly, also occurs in the mitochondria. Endoplasmic reticulum. Those are hollow tubes that allow transport and synthesis of the product. So it's not only like a passageway, like a subway tunnel, but it also synthesizes things that are utilized within the cell and synthesizes things that can be transported out of the cell. Okay, two types. You have rough endoplasmic reticulum or rough ER. That has ribosomes on the outer surface. It makes membrane proteins and proteins that are exported out of the cell. Smooth endoplasmic reticulum or smooth ER has no ribosomes present. It attaches to the ends of the rough ER and it makes products that are used inside the cell. Okay? Smooth ER has a lot of important functions. It makes steroids, it regulates calcium in muscle cells, and it destroys toxic substances found within the liver. Okay, ribosomes are known as protein factories. They contain ribosomal RNA and are made up of proteins and produce proteins. Okay, they make proteins by putting together or linking together amino acids by protein synthesis. So they take amino acids, because you know amino acid is the basic building block of all proteins, and then they attach one amino acid to another by protein synthesis. Golgi bodies are flattened sacs that lay on one another. So I try to explain it because you're like, okay, so you know grandma's purse? Grandma's purse is like huge. It looks like you can store a microwave inside of it. But when it's empty, it's kind of floppy, if you will. So imagine taking that floppy purse, that big floppy purse, and laying it on top of one another. That would resemble a Golgi body. It receives proteins, it modifies the proteins, and then they're put into vesicles for transport. So see those little circles? What it is those contain proteins in it, modified proteins. And I want you to think of a vesicle as a bubble, because it's a protective bubble that surrounds the protein, because if it's just placed in a watery environment, it's going to break down. It's not going to have the same potency or functionality. So it's almost like a capsule that surrounds Tylenol. The capsule will eventually dissolve, but it'll dissolve when it is time and when that medicine can be utilized best. Same idea. That's what a vesicle is. Okay, basically it receives changes in the proteins, it separates the proteins, and it transports the proteins where needed, whether it's maintained in storage within the cell or it is allowed out of the Golgi apparatus or move out of the cell. All right, so lysosomes, okay, they break down things that can be dangerous, quote unquote dangerous, like bacteria or cell structures that are failing. Think about, if it is not lined up perfectly, if there's certain things that are not functioning with the cell, it's going to slow the cell down. It's going to cause problems. It's not going to be able to do all the things that's necessary. So what happens? If we can get rid of those dying portions, those things that are not working within the cell, now it flees up the area to actually do its job. It contains enzymes. These enzymes break down things, and it's not the same enzymes as hydrochloric acid, but think about it this way. If something makes it into a lysosome, it has an enzyme strong enough to deteriorate, to break it down into something that will not harm the cell. Okay, so it's kind of like when things enter your stomach. It needs greater surface area, it needs to be broken down, so the enzymes found in that area would break it down to where it needs to be. Same kind of idea. It also lyses things. So the greater the surface area, the easier it is to break it down. If I lyse it or rip it apart, and then give enzymes to it, it's easier to break down than trying to break something down as a whole. All right, cilia and flagella, those are going to be things that are utilized for movement. They're made up of microtubules, okay, for strength, because if you think about microfilaments and microtubules, tubules are bigger, they're stronger. Okay, they move the cells itself. They also move fluid around, and they move. We've seen cilia on the surface of cells, especially when we get into tissues and you do it in the lab, you will see pseudocolumnar epithelium with cilia on the surface, so it allows things to move on the surface of the cell as well. Okay, so let's talk about the plasma membrane. I know I've already talked about it, but there's a couple more things that I wanted to note. Oh, there's that homeostasis award. I told you you'd be getting sick of me saying it. Homeostasis, remember, maintaining the inner environment no matter what the external environment is doing. That is that boundary. Remember, there has to be a clear separation between internal and external. Okay? So the function of the plasma membrane, what are its jobs? And I've kind of alluded to this. So it is a barrier. It is a clear separation. It does regulate what goes into and out of the cells, because things like oxygen and carbon dioxide move freely. However, there are those proteins present that can move things that are larger. It allows for cell recognition. with the carbohydrate tails that are found on the surface. Again, we talked about this. Anchoring sites, places where the microtubules and microfilaments can attach to give that shape to the cell. It allows for binding of enzymes. Remember, enzymes can catalyze a reaction, can speed it up. So this is there are places or markers on the cell surface, or receptor sites, which is a better term, that allow for enzymes to bind. It allows for cells to bind together. They're called junctions. And I want you to think of it this way. Cells sometimes fit together like puzzle pieces. Some cells, obviously, if there are two circular cells, there's no way it would bind together, but these junction sites help. And then it also contains that cytoplasm, because if there was a break in the plasma membrane, what would happen? That cytoplasm would come out. All right, so we've talked about cells, we talked about the organelles present, just the basic overall structures and functions of the different components of the cell. Let's talk how things move across that cell membrane, because we've alluded to it, and it's very simple. I'm going to keep it straightforward and nice and easy. So diffusion. Diffusion is the passive movement of something going with the concentration gradient. Going with the concentration gradient. That goes from an area of higher concentration to an area of lower concentration. And the idea is, if there's a boundary between the external and internal environment, if the concentration is higher on the inside of the cell as opposed to the concentration outside on the other side of that membrane, then what is going to happen is the substance is going to diffuse from internally to externally to balance it out. So when I talk about the passive movement or concentration gradient, think of it, I mean, I've just given you the idea, but think about it. If you are on a hill, if you go with the concentration gradient, going down the hill is significantly easier than trying to go up the hill. Down the hill really doesn't involve a lot of energy, but going up that hill or against the concentration gradient does. So simple diffusion, there's no energy required. It is with the concentration gradient, so no ATP utilized. Again, same idea. We just talked about diffusion. So simple diffusion means it's very easy. It's passive. Okay? And again, here's that whole diffusion idea. I'm trying to make it really prevalent for you because it's important for understanding when we start getting into some of the reactions the body does. So you can see from high to low. Osmosis is the same idea. It's the diffusion of water across a membrane. Same thing, high to low. It's just for water, we don't call it simple diffusion, we call it osmosis. Passive transport, okay, is simple diffusion. Again, not requiring energy, high to low concentration gradient, and it makes it through very easy. When we're talking about passive transport, it's just not diffusion. It's not just water passing through something as an osmosis. We're specifically talking about passage through the lipid bilayers without any energy required. It passes through easily. Remember, I told you the things that pass through easily with no help would be oxygen, for example. There also is facilitated diffusion. It's passive as well. It doesn't require energy either, but this is where we're going to use those proteins as like a channel or a bridge that allows things that normally can't just go through the phospholipid bilayer like oxygen, these are things that are much larger and need help. It doesn't require energy because they can pass directly through these integral proteins, things like glucose, things like amino acids. Active transport does require ATP. It requires energy. And notably, when it requires energy, it's going to go against the concentration gradient. Climbing that hill, it goes from areas of low concentration to high concentration. And we'll be talking about the sodium-potassium pump, okay? Though that sodium-potassium pump is active transport, it does require a lot of energy. Okay, so just keep that little note in your mind because we'll talk about the sodium-potassium pumps when we talk about the skeletal muscle as well as the nervous system. When we move things, we really want to move things in different ways. We want to move some of the big things. And one thing is exocytosis. Exo means to exit. Cytosis has to do with cell doing a function. Okay? So there's that transport vesicle that you can see. Remember, we talked about it with the Golgi apparatus, that bubble surrounding material. It goes all the way to the plasma membrane and it kind of pushes it out without damaging protein. It sends it out. Now it can move and do what it's supposed to do. Okay? Okay. Pinocytosis, pino means to pinch. So we're now going to take material, it's going to make it to the plasma membrane. The plasma membrane folds around it, making that vesicle bubble, and it leaves as a vesicle with a protein inside of it. So exocytosis, the vesicle opens, pushing it out without changing the substance inside of it. Pinocytosis means the substance itself makes it to the plasma membrane. It encircles it, it pinches it into a vesicle, and it comes off. Okay, endocytosis or phagocytosis is where it's kind of the opposite of pinocytosis. Something is going to come to the cell plasma membrane, and your body's going, or the plasma membrane is going to surround it and pull it into it, like bacteria. And I have this picture here. Here's phagocytosis. So at the very bottom of the screen, you can see bacteria. And there is the plasma membrane. Remember, it's fluid. It's going to come up, go around it, and pull it into the cell itself. Really cool. Take note, phagocytosis, I'm going to use that a billion times in this class as well. So make sure you understand phagocytosis. Exocytosis is large molecules being pushed out. And this is a great thing. There's that vesicle, there's that contents, and it pushes it out. Great depiction of what we're talking about. No damage to the plasma membrane, no damage to what's inside that vesicle. All right, now let's get into cell division. So this last tail end is to talk about a cell dividing, okay? All right, it is DNA replication. The idea is a cell has DNA present. However, I want to divide. So what happens? I want to take that one cell to go into two cells. So we have to equally divide the DNA into each of those daughter cells. So what is produced is identical to that original cell called a parent cell. Okay, there it is, that separation to be put into either. So it's separated, right? It's like pulling the ribbons apart. Remember, though, you have the adenine, guanine, cytosol, thymine. Well, it pairs up. It can make it, and then that original DNA, it's separated, it's transcribed, and now you have two strands of DNA. One goes into one daughter cell, the other goes into the other daughter cell, and it is identical to the original. So when we talk about mitosis, it only occurs in eukaryotes. That's plant and animal cells. It has four stages, which you'll see in a second. And it does involve the division of the nucleus. Division of the nucleus, meaning that the DNA is separated equally, it's identical in both daughter cells, is called karyokinesis. Okay, and there's the four stages of mitotic division. Okay, prophase, metaphase, ana, and telo. Okay, so prophase is going to be where chromatin in the nucleus condenses to visible chromosomes, which is the upper left-hand image. You can see these visible chromosomes. It looks almost like thread or fibers. And then the nucleus and nucleolus, you can't see them around it anymore. You just see these fibers present in the cell. Metaphase is this bottom left. That's where chromosomes attach to kinetochores. These fibers, they move them to the center of the cell. Now the chromosomes are lined up at the equator. Metaphase in the middle. Okay? Anaphase occurs quickly. Sister chromatids, now up in that metaphase, they're paired. They're called sister chromatids. And it makes sense. If I'm going to now go from that single cell to double the cell, I'm going to separate the sister chromatids. One's going to go to the left, one's going to go to the right, to opposite poles, not necessarily right or left. Okay? So you can tell it's anaphase because you can see those sister chromatids being separated. And just looking at that image, you know exactly where it's going to divide and become the two. The last one is telophase. Chromatids at opposite poles, you can see them. Okay? The spindle itself disappears. You can now see a nuclear envelope around the chromatids, and you can now see nucleolus. I can clearly say, yes, it's not, you can clearly see now two daughter cells. Cytokinesis, cyto-cell, kinesis is where it separates. Again, we've talked about it. This is the term for separating into two daughter cells. Now, it's done by cleavage foil or it's done by a plate. Okay, and there's the images. So it makes sense. So yeah, you're on anaphase and everything is separated, but we've got to separate it. And it's almost like it folds inwards. That's that cleavage furrow, which you can see on that bottom. Great picture. And you can see it where it's about to separate in that image as well. And then obviously it's not going to be the same in a plant. The plant kind of does the same, except for it makes a cell plate that separates the two.